All right. Good morning, afternoon, evening, everybody. My name is Jack Deckers, Iowa State University, and one of the co-PIs of the AG2PI program grants that we had. And this is our 30th field day already. And so the last couple of field days we've taken the opportunity to have researchers who were recipients of our last round of seed grants, what we call coconut grants, to talk about their grant outcomes. These coconut grants, they just ended just last month, and so we're eager to have them share their outcomes with the larger community. This is being recorded also, and as usual. So we have two speakers today. Fiona McCarthy from University of Arizona. She'll talk second, but we'll start with Dr. Daniel Runcie. He's an associate professor in the Department of Plant Sciences at UC Davis. And he and colleagues received a coconut grant that is entitled "When Models Are Too Good to Be True, Accurately Evaluating Phenomic Prediction as a Tool for Plant Breeding." So Daniel, I'll turn it over to you. And as usual, just put questions in the chat, and if it's immediately relevant to what the speaker is talking about, I'll interrupt the speaker, but otherwise we'll leave them to the end. So go ahead.
Okay, thanks for the introduction. So yeah, I'm just gonna jump in. So to introduce our team here, so my name, as Jack said, is Daniel Runcie, and I'm a professor in the Department of Plant Sciences here at UC Davis. And my main focus is quantitative genetics and statistics. The PI on our grant, the leader of it, is Mitchell Feldman. He's a new assistant professor in our department, and he's the director-elect of the strawberry breeding program. So he's taking over the strawberry program from Steve Knapp, and his main interest in what got us going in this project is that he would like to make improvements to how this strawberry program is working, and really interested in whether and how to invest in new technologies that could make the breeding process more efficient, more effective, and so on. And we're helped on this project by a graduate student, Fang Yi, who's in the Integrative Genetics and Genomics program here at UC Davis. So there are many possible gadgets and technologies on the market that promise better ways to measure plants. And so I'm just throwing up a few images with motivating examples. There are drones or handheld systems that use various imaging technologies to measure plants, usually in the field. There are big phenotyping instruments and chambers and things that can image plants on, say, a daily basis as they grow. And then there are all sorts of highly detailed tools for measuring the inner workings of plants, things like root architecture here, or molecular assays like gene expression or metabolomics. And these things can take thousands of measurements per plant. So each of these technologies promises in some way to describe the plants that breeders are trying to improve in all sorts of ways and know everything they can know about the plants. But among all of these potential options that, say, Mitchell could invest in, which technologies really are going to give the most bang for the buck? Which are the ones that are going to make his program the most effective? And the issue here is that many of these technologies are very expensive, and any investment that Mitchell makes in time or money in a new technology means a reduced investment in other aspects of his program. And so we have to be sure that they're going to help, or he's going to be worse off. Now, one way that we've tried to think about this is to put these various technologies on these axes here. So one axis would be how many traits per individual or per line can the technology measure. Call this one maybe phenomics technologies, versus the other axis is how many genotypes can be measured. Ones that measure more might be high-throughput phenotyping. So very roughly, we can place the platforms and technologies somewhere on these axes here, whether they're the technology or the measuring platform. But which of these dimensions is most important? Which one's going to give us the most use in terms of improving breeding? Now, I'm originally a biologist, and so these technologies in this part of the these axes sort of are the most exciting to me because they provide the greatest level of detail about the inner workings of plants. But what's sort of confused me for a long time is how can we really use this information in a practical way to make breeding decisions? Because that's not entirely clear. So yeah, this question has stumped me for a while. But Mitchell and I came across a set of papers over the last several years that proposed this exciting idea called phenomic selection. So there was this original paper in 2018, and then several others that proposed that we can use phenomic data in sort of directly in the selection process. So why is this exciting? So this is exciting because it's based in the idea of genomic selection. Now, genomic selection has been around for quite a while now, and it's very widely used and generally understood to be useful for improving the process of breeding. But the problem with it is that it's expensive, particularly that collecting the genomic data on enough lines is expensive and probably prohibitively expensive for many programs. So the idea here is that phenomic data, as I've described, at least from some technologies, can be collected a lot cheaper than genomic data, and has a lot of the same properties in terms of its scale and how it might be used. So the idea of phenomic selection is that we can predict, using this phenomic data, the sort of difficult to measure traits that we're interested in as breeders, using that phenomic data, just as we have been doing with genomic data. So the question is, can we use phenomic selection then as a cheap and effective replacement for genomic selection? Taking this program and using it to make our selections more effective. Basically, is it worth investing in these technologies that simply measure as many traits as possible on our target genotypes as cheaply as possible, and then plug that in to making better breeding decisions? Unfortunately, after studying this for a while, we've come to the conclusion that the answer is really no, at least not in this way. And there are two reasons for that I want to explain so that if you're going through the same process, maybe it'd be useful. So the first is that this comparison between phenomic prediction and genomic prediction that these papers are based on is really not a fair comparison, and is not really even the right question to ask. Instead, what we think is that phenomics can complement, but really can't replace genomic selection. And that's because phenomic prediction's use is in measuring traits, not genetic values. And so its use in the breeding process has to be different. So for the rest of the talk, I'm going to show you why we came to these conclusions. But if you have other thoughts, we'd love your feedback on if anything is, if we're missing anything. So as a brief outline, I'm going to start by explaining my view on how breeding works and discuss how genomic and phenomic selection fit into breeding programs. Then I'll show why comparisons between phenomic and genomic prediction are misleading and don't help us optimize a breeding program. And I'll end with a discussion of how we think phenomic prediction actually is very useful. So basically, at the highest level, breeding follows these steps. We start with a population of candidate lines. We measure traits on each of these lines. We use those traits to rank them, and then we select the lines with the best traits. Now, if these are good enough, we'll release these new lines as new products. And then we can also make crosses among them, and their offspring will then become a new population. Hopefully that population now will have better traits on average, so that the next time we go through this cycle, we'll have better lines to choose from and produce new varieties that are even better. These steps here constitute a breeding cycle. While simple, this process is very slow, mostly because measuring the traits well enough to do these selections well can take, in some cases, many years. So we're always looking for ways to make this process better. And using basic understanding of breeding, we can make this breeding better by changing two key parts here. First, rather than selecting the lines with the best traits, we should be selecting the lines with the best genetic values. By genetic values, we mean, in a sense, the average trait values of other plants with the same genotype. And second, these two parts here, really these two decisions that need to be made, really should be made separately and distinctly. The varieties that we release should be released based on what's called their total genetic value. which averages over things like environmental variation, G by E, measurement error, and so on. While the lines that we use to make crosses for the next population should be evaluated in a different way, based on something called their breeding value. Breeding values differ from total genetic values because they exclude effects like dominance and epistasis, because these are not well predictive about the quality of the next population. So it turns out that in terms of the long-term success of the breeding program, it's really about our ability to select based on breeding values to make crosses that's going to determine our success. So that's the part that I'm going to be focusing on. Now, all of this intuition that I've described here can be captured by this simple mathematical formula called the Breeder's equation. So the Breeder's equation shows that the rate of gain or success of the program in the long term is going to be the product of 4 variables: intensity, accuracy, the standard deviation of breeding values, and speed. Now, using this equation, we can study how any modification to our program is going to impact the long-term success by looking at the relations between these parameters. If we can improve one, that will improve our gain as long as it doesn't result in a decrease in the other parameters that will lead to the total product being smaller. So I'll go through these briefly because they're going to be relevant in what I'm trying to say. So first, intensity. Intensity describes basically the strength of the selection we're applying. And so it's the difference in trait value between the ones that we select and use for crosses relative to the whole population. We can increase intensity basically by increasing the number of lines that we're looking at. If we have a bigger population and we're taking the top 20 of them, the highest, that top 20 will have a higher value on average than if the population was smaller. Accuracy is basically how well we measure each of our candidates. In particular, it's the correlation between our measurement or estimation of their breeding value and their actual or true breeding value. Increasing accuracy by making this estimation process better is going to increase our gain. The next parameter is a standard deviation, which basically describes the amount of variability in the breeding values in our population. And so bigger, more variability means faster gain. But this one is not going to be very relevant to what I'm talking about, so I won't talk about it more. And then finally, there's cycle length, which is how long it takes between the creation of the population and when we make our decisions about which ones should be crossed to create the following generation. If we can make that decision faster, we're going to increase the speed, and thus we'll shorten our cycles and correspondingly increase the rate of gain. So given these parameters here, how do genomic selection and phenomic selection, as proposed, affect each parameter? And how do changes in one parameter affect the others? So we're using as a reference here genomic selection because it's well established as an effective tool for increasing the rate of gain in breeding programs. So how does this work for genomic selection? Well, first, so genomic selection is basically that making crossing decisions, the end of that pipeline there, based on information from genetic markers rather than waiting for phenotypes on each of the lines. So it turns out that we can show that genomic selection improves breeding because it can be used to improve the intensity, the accuracy, and potentially the speed of breeding. On the intensity side, as long as genotyping is cheap, if it's cheap enough, or at least if it's cheap enough relative to the cost of measuring each line enough times and enough fields that we get a really good value of it, then we can take that saved money to actually evaluate more lines by genotyping because they don't take up field space, and thus increasing our intensity. Genomic selection can sometimes increase accuracy also. When our target trait has low heritability, we can estimate the genetic values or breeding values of the alleles instead of the lines. And alleles in an experiment are going to be replicated more times than lines, and so we can have higher precision on their effects. And then finally, in terms of cycle length, the effect of genomic selection is that we can make our crossing decisions immediately without having to wait for many field trials, maybe many years of field trials, which reduces our cycle length and increases our speed. Now, putting this all together, it turns out, as shown by this paper here, that really the main benefit of genomic prediction or genomic selection is in its effect on the cycle length here. There are many breeding schemes where using genomic selection, you can cut out many years of your breeding cycle and correspondingly get great increases in gain. Okay, so that's how genomic selection affects the parameters of the Breeder's equation. But let me just go briefly through how it actually works. So using genomics in breeding, genomic selection, it relies on what we call genomic prediction models. Genomic prediction models are machine learning models that take in genetic marker data and use that to predict breeding values. So basically, what we do is we take a sample of our candidate lines, and we measure the target trait that we're interested in, say their yield in corresponding environments. And then we also, on each of them, we measure genetic markers, hopefully across the genome. We then take that genetic marker data and we fit it in some sort of a machine learning model to be able to predict that trait data. Then, once we have trained that model, we can then collect genetic genotype data on new lines that we haven't measured the trait on, plug those new genotypes in with the parameters that we've learned from the training side, and what we're going to get out of it are predictions of breeding values. We can then take those predicted breeding values, just as we have done the trait values or any other estimated breeding values before, and select lines based on that without having to actually phenotype them, thus saving the time, potentially increasing our intensity, and potentially increasing the accuracy of that. Now, the key idea here is that this genotype data is often very high dimensional. We might have hundreds to thousands to hundreds of thousands or even millions of different genetic features, which means from the statistical side, this is a P much bigger than N problem. We have a lot more features than we have lines that we're training our data on. But the neat thing about this is that we don't have to know which of these are useful. All we have to know is that there's some expectation that some of them are useful, usually because of linkage or pedigree structure or some other sorts of information that these markers are capturing for us. And if that's true, then these machine learning models, and there are many of them, ones like RR-BLEP or the Bayesian alphabet ones like, say, DAYS-B, or maybe more advanced machine learning ones like RKHS and so on, can capture information from these markers and use it to actually be pretty accurate. And so that's how genomic prediction models work. But the problem with them, as I mentioned, is that collecting this genetic marker data is still fairly expensive. So that's why the idea of phenomic selection was appealing. And it's the basis of this idea that phenomic selection could possibly replace genomic selection. So the idea of phenomic selection, I'd say, is sort of deceptively similar to genomic selection. Also, we start by building a model, a prediction model. This time I'll call it a phenomic prediction model. We use basically the same procedure. We're going to take a sample of our lines. We're going to measure the target trait, the one that we're actually interested in, on those lines. And then rather than measuring the genotype on those lines, we'll collect some phenomic data. We're going to collect a set of phenomic features on each of them. These features might be various wavelengths from a near-infrared scanner. They could be those wavelengths or other measures of traits, say at multiple time points collected by a drone flying multiple times in the season, or they could be multiple traits in multiple different environments. But either way, there's going to be a bunch of different features that we collect, hopefully in a high-throughput way, in a cheap way. And all we're going to do is we're going to take those as a matrix and then plug them into this same type of same sort of a prediction model. Once we've trained it, we can then use that model by collecting that same cheap, high throughput phenotyping data on a set of new lines that we don't have the target trait on, take those features and plug it into our prediction model in the same way that we did for genomic selection, get out these predicted phenotypes, and then the idea would be to select lines based on those phenotypes without having to measure the target trace itself. So the motivation here is really very similar to genomic prediction. Again, the phenomic data that we're interested in is often very high dimensional. p greater than n. And again, we don't really know which of these phenomic features are going to be most useful. So we just throw all of them again into our model, and we hope that some of them are going to sort of stick because of genetic pleiotrope. And it turns out that a lot of the same very simple statistical machine learning models work very well for this process. And so that's nice because it means that in practice, it's very easy to get a phenomic prediction pipeline up and running in your system. You can basically plug and play and get these models working very quickly. And so that's what the papers that I showed earlier did. And they provide data like this, showing that these phenomic prediction models can be competitive with and often beat genomic prediction in their predictive ability. So here I'm showing a data set from Rincent et al. In 2018. In the yellowish colors are several phenomic prediction models. This is wheat data, either heading data or grain yield. And then in the blue models is the predictive abilities of the corresponding genomic prediction models in these same environments. And what we see is that for many of these phenomic prediction models, they actually have higher predictive ability than the genomic models. So the argument that they're making here is that predictive ability is related to the accuracy in the Breeder's equation here. And so what this implies is that phenomic selection may actually increase the rate of gain relative to genomic selection at a fraction of the cost. But after studying this a bit, Mitchell, Fang, and I have come to the conclusion that this is premature for a couple of reasons. First, this is not the right measure of accuracy. And second, the parameters of the Breeder's equation are just too interrelated to be evaluated independently like this, to only look at accuracy itself. And as I mentioned before, if we're comparing to genomic selection, that's optimizing other parameters in this equation more effectively than we can do in phenomic selection. So I'm going to show what I mean for each of these. But first, to get some audience participation, I'm going to run a poll. The question that I'm going to ask is this. Do you know what the difference is between predictive ability and accuracy? Have you heard that before? Because it was actually a term that I had not heard until fairly recently. It looks like responses are slowing down. Give you another couple seconds. Okay. I'll cut it there. So the results are about 2/3 saying no and 1/3 yes. And so that's more or less where our group was when we started. These words, predictive ability and accuracy, sound very similar. I'm going to close this. Sound very similar, but they're not the same, and they're different in very important ways that are relevant to this question of comparing phenomic and genomic prediction. So starting with accuracy. Accuracy, as I defined before with the Breeder's equation, is a correlation between our predictions, whatever model we're using, the predicted value we get out of that, and the actual true breeding value of the lines that we're trying to study. Now, this is the number that's in our equation, but the problem with it is that we don't know what the true breeding values are. We can never know this with certainty because it's sort of this theoretical concept. And the consequence of that is that we can't measure accuracy. We can't know what the actual accuracy of our model is because we can't observe the breeding values. So a statistic, though, that we can measure is predictive ability. Predictive ability differs from this in that it's the correlation between our predictions and observed trait values. Now, we know our predictions, they come out from the model, and we know the trait values because we've measured them. So this is a number that we can measure and can report. But the thing is, these are different numbers. They have different values. Now, that would be maybe okay if ability was an unbiased estimator of accuracy, but it's not. For genomic prediction, we know that ability is always lower than accuracy. Now, even this bias here, even if ability, as we can measure, is always lower than that, if that was the same relationship with phenomic prediction, we might still be okay. Maybe not perfect, we don't know exactly the rate of gain, but we are able to compare methods appropriately. But that's not the case. For phenomic prediction, sometimes ability underestimates accuracy, but sometimes it overestimates accuracy. And that means that the relationship between ability and accuracy is different for genomic and phenomic prediction, which means that we can't compare them. Now, the reason for this is that the observed traits differ from the breeding values because they're noisy, basically noisy estimates of breeding values. And if we just take some basic ideas from quantitative genetics, we can write this noise has various components. Might have non-additive genetics, microenvironmental variation, measurement error, and things like that. And while these genomic and phenomic prediction models may look the same and are often written like this using the same symbols, they're really fundamentally different. In genomic prediction, once we've trained our model, when we then use it to predict the breeding values of new lines, because of the way that we've parameterized, we've put in our genetic marker data, this prediction really only contains information about their breeding value. But in phenomic prediction, when we train our models, the features that we're using, those features themselves contain information about non-additive genetics and often microenvironmental variation, which in effect contaminates the prediction ability relative to accuracy. It's these non-additive genetic and microenvironmental effects that make the phenomic prediction successful in a sense, but they make our predictive ability measure differ more from the actual accuracy. Now, the way that we usually measure these things is using something called cross-validation, which is in a sense supposed to prevent overfitting like this. But something that I showed with Hao Cheng a couple of years ago is that this type of contamination in a phenomic prediction model is not removed by cross-validation. Cross-validation estimates are just as bad as any other way of estimating the accuracy of these models, if that's what your goal is. Now, just a couple of side notes here. This statement that genomic prediction only contains information of the breeding values of new lines is true if we're using or approximately true if we're using models like R, BLUP, and Bayes-B, simple additive models. But if we use one of these newer machine learning nonlinear models for genomic prediction, that's going to have information of non-additive genetics, and so it's going to be contaminated. The prediction abilities are going to be contaminated just like they were in phenomic prediction. And in phenomic prediction, the contamination from non-additive genetics and environmental only applies if our validation data, the observed traits we're looking at, are measured on the same plants as the phenomic data is collected on. But some applications of phenomics use something like a platform or a chamber to measure phenomic traits. And so in that case, there isn't any contamination of environmental variation, but there is still going to be contamination of the non-additive genetics that's not part of the accuracy, and thus will still bias our answers. So here are some graphs showing the magnitudes of the problems I'm talking about. So let's start with genomic prediction. As I've said, in genomic prediction, accuracy is always greater than ability. So in each of these graphs, I'm showing 3 settings here that differ in the heritability of this trait that I'm trying to do selection on, from low to high heritability. And on the x-axis, I'm showing different predictive abilities you might measure, and then y-axis being the corresponding accuracies of the model. And so the black line here is 1-to-1, and we see that in all scenarios, regardless of the heritability and regardless of the predictive ability you measure, accuracy is always higher than ability. And the relationship is pretty simple. Accuracy is equal to the ability divided by the square root of the heritability. So that means that when the heritability is low, the underestimation is even greater. When the heritability is high, accuracy and ability are pretty closely related. But that gets a lot more complicated for phenomic prediction. So I'm showing here the same 3 scenarios. again, for different levels of heritability of the target trait. But now for each scenario, each of these lines shows a different level of contamination of, say, non-epigenetic and microenvironmental variation in that phenomic prediction ability estimate. So the way that I want you to look at these graphs is to take a specific level of ability. Say we run a phenomic prediction model and we observe that our ability is 0.6. What are the possible ranges of values of accuracy? Well, if the heritability is high and our ability is 0.6, accuracy might be as low as about 0.3 or as high as about 0.7. But if the heritability of our target trait was lower, say 0.4, then accuracy, the actual accuracy might have been as low as almost 0 or as high as 1. So in here, and it gets even worse if the heritability is even lower. So basically, predictive ability tells you very little about the actual accuracy. Regardless of your predictive ability, your accuracy could range from -1 to 1. So really, it's telling us almost nothing. And this gets worse whenever the contamination due to G plus E is strong, or whenever the heritability of the target trait is low. And these are the settings where we might have been most excited about using phenomic prediction. So is this contamination actually enough to matter? Well, what we did is we went back to several of the papers that have proposed phenomic selection and reanalyzed their data again. And this is the work that Fong has been doing. And so here are two examples from the original paper, an example of predicting grain yield in wheat in a specific condition, and another example of predicting the circumference of poplar trees. And so here, the two bars, the red bars are showing the ability of a genomic prediction models, and the blue bars are the abilities of a phenomic prediction model. And so the observations were in both data sets that phenomic prediction beats the genomic prediction if we're scoring them with predictive ability. So what we've done is we've developed an R function using some existing theory that shows a way of directly measuring or modeling the accuracy, not using the direction observed ability, but trying to model the accuracy of each method. We're turning this into an R package, which isn't quite ready, but will be around shortly. And so if we do this and try to estimate the actual accuracy in each of these cases, we see that in both cases, the accuracy of the genomic prediction model is much higher than the reported ability. In the grain yield, the accuracy of the phenomic data was actually pretty good, but for in the poplar, the accuracy is actually lower than ability. And across the various data sets we've looked at, phenomic prediction rarely beats genomic prediction in most data sets in terms of accuracy or predicting breeding. So that's a little disappointing. But stepping back a bit, does phenomic prediction actually need to be more accurate? Remember that the parameters of this equation are interrelated, and it's only the product that matters. So even if our accuracy was lower, if intensity could be increased enough to compensate, then maybe phenomic selection schemes might be useful. And because phenomic prediction is cheaper, or the phenomic data is cheaper, maybe we'd have more money to be able to increase intensity. But how much would intensity have to increase? How much would population sizes have to increase to compensate for any loss of accuracy? So we studied this, and unfortunately, it turns out that you need tremendously larger populations. So here, the answer depends a bit on how big your population starts with. But say we're starting with a population of 300 lines that we're evaluating by genomic, that we could evaluate by genomic prediction. If we doubled that to 600 lines, we would get a gain in intensity of only about 10%. If we went tenfold higher to 3,000 lines, our gain in intensity would be about 40%. And even if we went 100-fold larger, we would be only getting maybe 70% or so gains in intensity. But as I said earlier, genomic selection can easily get 100% gains by shortening cycles. Say in half, you'll get a doubling of the gain. And so it's really hard to see how phenomic selection could be used in a way that could compete with genomic selection at all in terms of the overall rate of gain, because it doesn't have the ability to shorten cycles in nearly the way that genomic prediction does. So we don't think phenomic selection is the optimal use of phenomic data. But what is? Well, according to our study of the Breeda's equation here, we really want to be doing genomic selection. We want to be doing it because it's best because of the speed. So could we use phenomic selection to improve other aspects, though, within a genomic selection framework? Well, genomic selection is still going to be expensive because of the cost of genotyping, and that's going to limit our intensity. So we can't make any improvements there, but maybe we can make improvements in accuracy. Genomic selection relies on these genomic prediction models, and the accuracy of them is limited by training data quality. Can we use phenomic prediction to get better training data for genomic prediction models? Well, the training data quality has two components: its size and the heritability of the trait. The training data size is still going to be limited by the cost of genotyping, so we can't make any gains there. But to increase heritability, we can measure each line for that target trait more accurately. We can measure it more times, or maybe even better, we can measure related lines more times. And so these are things that are discussed by Lane and Murray, talking about using phenomics and high-throughput phenotyping to increase heritability in a very cost-effective way. And so we think that this is the way that phenomic prediction models should be used. And interestingly, if you're using the model in this way, it's actually prediction ability that we care about. The correlation with the target trait is the right way of measuring the success of a phenotypic prediction model with respect to the goal of increasing the heritability on a line mean basis. So back to these results. I've argued that for genomic prediction, we really want to be focusing and reporting the accuracy of the models, not the abilities. And that takes a little bit more work, but it's the correct measure to predict the genetic gain. But when we're doing phenomic prediction, we really should be reporting, as is done, the ability of the models, correlation with Y, because our goal is to improve the heritability of the traits. And so to improve the heritability, we need to measure the traits better. And so my final point here is that these numbers really shouldn't be compared. We shouldn't be putting them on the same axes because they're measuring different things. And so the comparison between them is misleading and is really not useful itself for optimizing breeding. So to summarize, we're excited about high-throughput phenotyping and phenomics, but realize that they're expensive. Phenomic prediction as a statistical approach works great, and it works a lot like genomic prediction, and a lot of the lessons that we've learned from data handling and such can apply, but it really needs to be used differently in breeding programs. We want to use it to measure traits, not breeding values, and we want to report its accuracy as predictive ability, not predictive accuracy. So those are my thoughts on this topic. I'd like to acknowledge a few other collaborators that have discussed this with us and various funding sources, including AG2PI, the California Strawberry Commission, High-Phen Phenotyping Systems, and the Department of Plant Sciences. And I'd be happy to take any questions.
Okay, thank you, Daniel. Very interesting presentation, interesting work. And yeah, I mean, this whole area of accuracy, predictability, there's a lot of confusion out there. And it's important to get that the distinction made and clarified and use things in the proper way. So there's some questions in the chat already. So Anya Walsh asked, to gain phenomic data, you need to create individuals or line crosses and measure them. For genomic prediction, you can in principle predict the performance of lines that don't exist yet and which crosses you should make to maximize the probability of top performance. Is that correct? Is that the correct understanding of the difference?
Yes, I think that for phenomic data, you have to have a line existing to be able to measure any of its phenotypes, so any of its phenomic traits, and so you'd have to wait there. And while, yes, genomic prediction could, in principle, be used to predict the outcome of crosses, and this actually is another of the projects that Feng, the graduate student, has been working on, evaluating within the Strawberry breeding program here, how successful genomic prediction models might be at predicting actually what we're interested in is the variability, the amount of variance that a cross will produce. And so whether a particular cross might produce extremely good individuals. And so another criteria that you can use besides the breeding value is called the optimal cross value. Did I get that number? Maybe, or the usefulness criterion. There's various things that you can do to try to predict maybe multiple generations ahead. And again, yes, this is something that genomic prediction can do and phenomic prediction can't do. And so another reason why they're not really compatible.
Yeah. It's really, predicting the phenotype versus predicting the breeding value, and the breeding value we need for genetic progress. So there's a question from David Gonzalez. So phenomic prediction for variety release, because it contains non-additive genetic effects, but in contrast, genomic selection should be used for selecting parents. Is that the right interpretation?
Sure, that makes sense.
Yeah, and that's what I was thinking, that if you because you just differentiate between identifying the best varieties to release, so that's where you're interested in the total genetic value, and then capturing the non-additive genetic effects is what you want to do along with additive, correct?
Yeah, assuming that your release can capture the non-additive genetics. So if you're releasing an inbred line, then yes. If you're releasing a population with some species, you won't be able to capture the non-additive genetics in that release, and so you wouldn't want to actually over predict in that case. But generally, I think that in that distinction that I made between total and breeding value, then yes, phenomic selection is fine as a tool for evaluating things in terms of their total value more accurately, because you can do it more times and more environments with more precision.
Yeah. So there's a question from Seth Murray. I'm not clear on how you determine that genomic selection has a 2x rate, 2 times as high a rate of genetic gain. Says that I have not seen the actual results near that high. Isn't it only fast if you have seed chipping technology and can pre-select what is planted? Similar on genomic selection, isn't there greater potential for a genetic bottleneck or erosion when you use genomic selection?
So the two separate questions there. The first one is, how do they come up with that 2x rate of gain? And I just so I think the 2x approximately. Well, I guess I was thinking in a case where it takes you 2 years to phenotype, or even 1 year to phenotype. If you can genotype much faster, if you can genotype in half the time, then you do that. And I think there's lots of breeding systems that take many years to collect enough phenotype data to make crossing decisions. Genotype data could be collected. If it can be collected in half the number of years, you get a half rate of gain. My point really is that I think that in most breeding systems, there's possibilities of fold changes, multiplicative effects, or greater than 100% changes in speeds, in cycle lengths by evaluating earlier. And there's really not the opportunity to get that scale of change by modifying intensity or accuracy or variance.
Yeah, and that's what that's what we've also seen in livestock, in dairy cattle. Genomic selection has allowed a large decrease in generation interval, because we don't have to wait for the progeny test on the bulls, and we see a doubling of rate of genetic gain. So okay, so well, there's more. Go ahead.
No, that's fine.
There's more questions in the chat, so maybe if you can address those in the chat, and then if there's time, we'll come back to them also if after the next presentation. But Daniel, thank you, and I'm going to turn over the floor to our second speaker, who is Dr. Fiona McCarthy. She's a professor in the School of Animal and Comparative Biomedical Sciences at the University of Arizona. So also a recipient of one of the Coconut Awards, and she is going to talk to us about developing bioinformatics workflows to support agricultural genomics. So Fiona, floor is yours.
Thank you very much, Jack. So As Jack mentioned, this is a complete change in topic. We're talking more about developing these bioinformatic capacity. And really, what I want to take you through today is I guess, like many of these short-term projects, a work in progress. So our rationale for this is, of course, we all know that it's much easier to generate data than it is to analyze and understand the data. And I think that's true of nearly all data types we're using in agriculture, not just the genomics data. But one of the things that we have very often in agriculture is those silos between the plant and animal sciences. And I think that bioinformatics is very often a good way to bridge those silos. So what we were thinking about is that even though we're using the same types of data sets and analyzing the same types of sequence data, how are we doing this the same? And can we, develop these opportunities for plant and animal scientists, researchers, and specifically perhaps the bioinformaticians to, figure out how we can more effectively use the resources that are available to us. And part of what we wanted to do here was look at this in terms of our analysis workflows. Perhaps there's ways that we can develop these workflows that, can be used to help the community. We understand that everyone has their favorite way of analyzing data. We're not here to tell you that you can only use this software or this workflow, but rather by providing these, we provide a toolbox, if you like, for other researchers to start getting in and analyzing their data. But also we provide a set of well-documented and tested workflows which can be used as benchmarks and also be used, and by that I mean, I guess, benchmarks to test similar workflows and look at the output accuracy and things like that, but also to have these available for education and training. So really what we're doing here for this project is developing the collaborations between plant, animal, and also microbial bioinformaticians, to support the agricultural research that's going on. And the second part of this project is really about, can we work on developing some workflows which would, take commonly used data types or do commonly used analyses, and have these, really well-developed, well-documented, some example data sets with them, and develop these for community use. And a third outcome of this project is that we're really looking for the people who are currently, spending a lot of time, trying to analyze data, and that's our graduate students, obviously our postdocs as well, but this project focused on the graduate students. So what we wanted to do was to support graduate students who were already working on doing these types of analyses to, take a little extra time to think about how to document and get that workflow public so that other people can reuse those workflows. So for our first goal, we were really trying to get this discussion of agricultural bioinformatics needs going. And I will say that my co-PIs, Pankaj and Stephanie, and I were enormously assisted by the wonderful people here. We decided we would have a workshop at the 2024 Plant and Animal Genome. So we developed this workshop. It wasn't a traditional workshop where everyone speaks, but rather we had this group of panelists, and we led a discussion around this idea of what is it that the community needs. So I really want to take a moment to acknowledge these people. They did a phenomenal job. Sushma Nathani from Oregon State. She's got many years experience in working with, taking genomic data and biocurating it into public resources for people. She's also done some phenomenal work engaging students in parts of that. Kristen Berlew is, she's a researcher with ARS who's also associated with the University of Missouri. She is a plant researcher who doesn't have as much bioinformatic expertise, so her point of view was enormously helpful. Ted Kalbflash from the University of Kentucky and James Coltes are both bioinformaticians working in animal genomics. And Millie Woku from North Carolina Tech, and she is one of these fabulous animal science researchers who thinks about these things and thinks about it in terms of education. So we were trying to get this group of people who could really sort of think with different perspectives, who could address this. In addition, on this workshop, we were lucky enough to have some graduate students and postdocs who turned up as well and were contributing to this discussion, which was really great to see. But in conjunction with our panelists, we really came up with these 6 questions. I'm not going to read them all, but they really were trying to focus around this idea of what is it that our community needs, and how do we go about getting to that point where we're addressing these needs. I will take a moment to say that these are open-ended questions. We're writing a report and trying to get that information that we got all synthesized. We've set up a quick survey. If you're interested and you want to contribute to this, we're still collecting that information. So that link is available, and it'll be on my next couple of slides, and I'm hoping that we'll also be able to put that into the chat for you if you're interested. It literally is your opportunity to contribute to any of these questions that you are passionate about. So I want to go through some of the initial findings. I'm not going to present everything, but more or less highlight some of the points where we spent a lot of time discussing this. The first point about what are the barriers that the research community has to doing these type of analyses. I was interested to see that access to computing still comes up as a problem, despite, we've had some major funding to push towards efforts to try and address this. So that's interesting to know. The much discussion of poor documentation. This, I think, is a reproducibility problem. But more than reproducibility, it's also this idea that, sometimes these software manuals and protocols are written without enough detail, or perhaps at a level where not everyone can understand and follow the process. And I will make a point here of mentioning that James Coltes is and his group have been working on developing a data reuse white paper, which talks about reproducibility. So I'm not going to go into a lot of detail about that, but I think that's going to be a really important piece of this puzzle of how we can better support our research community. We also wanted to have everyone take the time to think about what plant and animal scientists have in common, because we too often spend a lot of time thinking about what we don't have in common. And I think that's a good way to think about some of this discussion. We do have a lot of these same barriers. We are doing the same types of analyses, generating those same sequence data that we done. And something that came up repeatedly is that the software that we're using is mostly designed to work for non-model organisms. It always will take a little bit of tweaking to get it to work on the species that we care about. So I think that's a good way to think about that. We also discussed this idea of how do we make sure that we're training the next generation? How are we making sure that our students are going to have the skills that they need to continue to do this? And this discussion, I think, was really interesting. We spent a lot of time talking about this. It was great that there were graduate students in the room. A lot of these ideas probably not novel, but it's interesting to hear that they continue to come up despite, you know, efforts to working towards this. So we haven't addressed these yet. Again, I'm not going to talk in a lot of detail about this. I think this is going to come out in the report in a lot more detail. And the final question, and I think this was something that, again, we spent a lot of time on, you know, what is it that we need? And what do we need right now versus what do we need in, you know, the medium term future? And again, we see these types of things coming up. This is kind of a synthesis of these, going back to that idea of what are the barriers. But I think it's important that we address, you know, it's not just enough to point out the problems. We've got to think of ways of addressing them. So it's interesting to see that there was a lot of discussion of how do we get these workflows and make them accessible for people who don't have a lot of bioinformatics training. And I think that that segues very nicely into the next goal of this project. And the next goal of this project was to develop some accessible workflows, and not just develop the workflows around very specific sets of data, but to make sure that these workflows are accessible, that they're well documented, that they have worked examples with public data sets as examples, which hopefully then we can move on to then taking this workflow and applying it to our own data sets. So we provided funding for graduate students. And the idea here is that, you know, the graduate students are very often working on analyzing their own data. And, you know, there's a push, you know, to get that data analyzed and then to look at it. And what we wanted to do was to give them a little extra time and support to help really think about the workflows that they're developing, you know, get the documentation really well organized, and to get that online or publicly accessible. And so we did some public calls. You would have seen emails going out from AG2PI and every other mailing list that we could think of, and the students were invited to apply for this. They were selected based upon the type of workflow and whether that's something that would be commonly used by the community. Whether the data type they wanted to test this on was publicly available. And then along with that, you know, whether they have a good history of making, in their research lab, of making workflows and data sets publicly available for the community, and they have the support to do that. So that was the first part of the goal. The second part of this was we wanted to bring in undergraduate students to, first of all, try and reproduce these results just using the documentation that was available and seeing if that was possible. And that's a great way of providing feedback to the graduate students about how they can adapt their documentation to make it more usable. But then the second stage of undergraduate participation here was for to identify data sets from different species and to bring those and to run a different data set on that workflow and see how that stands up and whether that's still working well. I'd like to take a little time just to go through these workflows. And I should say too that, you know, these links that we're providing will all be available. So if you're interested, you should be able to access those. The first student was Kyle Parker. Kyle Parker's a graduate student at Texas A&M, and his workflow focused on variant calling. So what he was doing here was taking the next-gen sequence data, aligning it to the reference genome and producing a variant call file. So this is a really nice example of a workflow that many people would want to do. This was designed to handle wheat, and so we're going to be interested to see how that stands up when we start looking at other genomes and other data sets. He's made all of his workflow available on GitHub at this link shown here. And he's also provided documentation through a README file. And so that's one of the workflows that are available. Our next student, Kaylin Rayleigh, is a graduate student at Cornell University. And she's working on predicting antisense long non-coding RNAs. So what she's doing here is she's using both short and long read sequence data and looking at developing transcript assemblies, and then she takes it through many different steps to do, not only to do the long non-coding RNA identification, but then to select those long non-coding RNAs that are specifically antisense. So she has a long list of steps that she does. I haven't given all the details here. She initially used this on some plant species, some brassica and cannabis species, and as part of her graduate research. And again, she's provided the GitHub link where she's put all of her scripts and provided documentation for this so that you can work through this example as well. Our next student is Yushi's from Nebraska-Lincoln. And he's been looking at RNA splicing variants. So what he's done here is he's been looking specifically at RNA splicing variants from RNA-seq data. He's also added an additional piece to this where he's looking at microexons, those very small exons, and how they contribute to alternate splicing. So this is a very interesting project. So if you're interested in this kind of thing, again, it's available on GitHub. He's put his workflow on GitHub, and again, the README documentation available with that. This student, Mustafa Shokroff, is located at University of California, Davis, and I should actually say he's recently graduated, so congratulations to Mustafa for graduating. His work has focused on structural variants, so he's doing this very interesting approach of combining long and short read data sequences and using that to detect structural variants. He initially used this on cattle genome data, and again, this idea that we're hoping that we can expand this out to other species. Again, it's available on GitHub along with the documentation. Gustavo Chinetti is based at Virginia Tech, and he's got a very interesting project where, again, this idea of integrating different sequence data types. So he's integrating the RNA-seq data with nanopore data and trying to identify transcripts, in particularly looking at identifying potentially novel transcripts. His approach looks at both doing de novo assembly and reference-based assemblies for genomes. And he also makes sure that he's comparing his results to both NCBI and ensemble annotations, which I think is important because we do know that there are differences in those annotation sets. And the other thing his workflow does is he looks at these novel genes and he works to classify these as either coding or non-coding. So this, I think, is another example of a really nice workflow that could be applied in many different ways. I've given you the GitHub link here, and again, we've checked to make sure that the documentation is also available with that GitHub link. Kylie is a graduate student at the University of Kentucky, and this is a slightly different project. It's not a workflow that's portable and you can pick up and use, but rather it's a querying tool. So this, you can think of this more as a process for thinking about how to do, how you might make your data more accessible. Okay. So this, what this does is it makes these large genetic data sets directly queryable from their location, say at your home institute. So it allows your researchers, in this case, it might be your collaborators, to set up these remote queries and run these remote queries quite fast through large data sets. So the example here that they used for testing this was they had RNA-seq data across 4 animals and 7 different tissues, and they were able to do the indexing and pull out specific queries which they used to generate a PCA plot. And I think that whole process was done in, if I remember correctly, in a couple of hours. So this is a really powerful method. The documentation is all available, and I strongly encourage you, if you're interested in this, to be really working on sorting through large data sets, to contact Ted Kalflash at University of Kentucky for this. So those are our student projects. I'm really excited to see what the students were able to do, and I think one of the things that we want to do is to keep building on this. For our ongoing work, we're obviously still writing up our discussion of our community needs for how bioinformatics can go on to keep on supporting our agricultural research communities. And we're still putting that together, and of course that's going to be made publicly available as soon as we can. So I encourage you, if you're interested or if you have something to say about that, this website allows you to fill in comments about any of these questions that I showed you before. The graduate students have made their workflows available. We're still in the process. We were delayed on the start of this project, so with that came the delay in this process. So we have a course of undergraduate students here at University of Arizona. They work in it's a bioinformatics course where they get some hands-on experience developing small research projects. So the idea is that we're going to have them test this documentation, test these workflows with the data sets available. And then ideally what we'll have others doing once we get that feedback is the opportunity to make those changes and finish up by having a second test run where where we select new data sets from a different species and see if that workflow is robust for that. The other thing that we've been thinking about as a group is how we can continue to support our graduate students in this. I think we've learned a lot from doing this project. I owe a debt of gratitude, not just to the graduate students, but to their mentors who've been through a pretty grueling process of administrative hoops to jump through from different institutes to try and get this funding support to them. So I will finish up by thanking all of the many people who've been involved in this project. I owe the funding support, of course, comes from AG2PI, which is supported by USDA NIFA. I really owe a great debt of gratitude to my co-PIs, Pankaj Jayaswal and Stephanie Mackay, who've been really interactive and helpful throughout this project. I thank the people who agreed to step up and serve as panelists, and more importantly, to everyone who came along to this workshop. Again, especially to the graduate students and postdocs who not only showed up, but spoke up. And it's great to get those different perspectives. And as I just mentioned, I want to finish up by thanking not just the students, but their graduate advisors who had to put up with an enormous amount of emails from me and my administrators to make sure that we got everything lined up and organized so that we could do this project. So I'll stop there, and I'll take this down so that I can see the questions.
Okay, thank you, Fiona. Very impressive.
Thank you very much.
And great to get the graduate students involved. And I think we also have, listening to both of these presentations, I think we also have to realize how little time they had. The timeline for these projects was very short because of the funding limits, boundaries. So yeah, impressive what has been accomplished. There's a number of questions in the chat. Let's see. Where is it? Okay. So one of the questions was about getting more details on the workflow on the variant calling, and you gave the link. So what I'm suggesting is, yeah, to go. I think that's the purpose, is to go to the link. It's not so much about what is exactly that workflow, but the information is there. And then if there's questions, I think they can still contact you, right?
Yes, and if you have any trouble finding this workflow, but I'm going to make all of this available on the website. But if you have any trouble, please reach out to me. I'm happy to connect you up with the link or the people who develop these workflows.
Yeah, yeah. Okay, then was a question. My impression is that the ensemble improves annotations of the data they consume. I'm not sure. From NCBI, so I'm not quite sure.
Yeah, I can talk about this because I've actually looked at this directly, and what we've done, and this was done because of my personal bias with the chicken genome, and I can tell you that there is not one that is better than the other. What it is that each one is better at different things. And I can tell you that over the last few years, I have seen those assemblies starting to move together. I don't know whether that's a good thing or not, because we don't know whether they're moving, they're in more agreement, but whether that agreement is correct, we don't know. Yeah.
Yeah.
So.
Okay, next question. And actually, that's a question I had too. I can see great value in developing robust, well-documented workflows, but is there a risk of creating, as an unintentional consequence, copy-pasters who do not understand necessarily what they're doing, and if the results make sense, but if you get numbers, got to be right.
I think that we have that in any case, right? Because we've got people out there who, you think about any of those online tools, and I'll pick on, functional enrichment, Go enrichment. You can throw in any random set of genes and you'll get something. Whether it makes sense or not is a different story. I think that we need to make these the ability to analyze this data more accessible, because we don't have enough bioinformatics people to do everything. What we need to do is to really develop those skills and bring the capacity to make it easier for new people to access that genomics data. I don't know if that's a reasonable answer, Jack. I know there is that concern about, the copying and pasting, but I think this is something that is already being done, essentially. So that's not something I worry about from that perspective.
Well, but shouldn't we, in addition to teaching our students to use the tools and develop the tools, when they use them, should we also teach them to critically look at the results and do a lot of checking, a lot of validation to make sure that they're getting what they think they should be getting?
Yeah, one of the things I would like to see in the documentation, and I don't think you get that on a first pass or even a second pass, is, a list of, I've reviewed so many papers where every when you get to the bioinformatics part of it, it's this software, this software, this software. It's all run at, whatever out of the box parameters were. So one of the things I would like to see in workflow documentation is this analysis of these are the parameters you should be looking at in these types of situations. And, that's essentially what we're trying to distill and get to our students when you think about, critically analyzing and adapting workflows to work for different situations. So, but that's sort of passed down verbally and it's not written down. And that's what I think we need to see in our documentation.
Yeah, no, that's a good point. Yeah, yeah. Yeah, keep the questions coming. I think I've exhausted the chat question. I have one. So if I have a problem and a bioinformatics problem, how do I find the right tool?
This is, I don't know, there's a couple of things here. So first of all, what is the right tool, right? So very often when someone has their first set of genomics data, there's a question of, well, now that I have this, how do I analyze it? Some people might look at the papers and say, well, they did this, so I'm going to analyze it the same way. But and that's where you create this idea of, no, I can only analyze it this way because that's what we used 20 years ago. and you don't get that change. But I think there is no right tool. I've spent a lot of time in different stages of my career where I've picked out 6 different tools that do the same thing, analyze the same data set, all with those tools. And I think the thing that is more important is to understand what's working underneath the hood of those tools and what assumptions are being made. And that can be very, very hard to tease apart. But it remains a problem. We need to see that transparency that we don't always see in tools. And the biggest problem, I think, is with commercial tools, where you start trying to look at this and ask those questions and you get a lot of pushback. So I know that doesn't answer your question, but I would say this is going to vary by data sets. So I think what you should be doing is looking at least a couple of different options. And don't assume that the software you always use is the best option, but also looking at the parameters, right? You shouldn't just be running it through with default parameters. I think that is something I'm much more concerned about.
So should there be an effort to compare different workflows that aim to do the same thing?
Yeah, and this is what I get at with the benchmarking. We haven't really developed it, but this idea of, if we have a workflow that we know works well and is well documented, and we know it's documented against at least a couple of data sets, then that gives us a comparison point. Now let's use that and use different software to do the same thing, and then look at those comparisons. What do those outputs look like? Does that make sense?
Yeah, no, that's right. I was so for those online, you can put things in the chat, but also raise your hand if you have a question, and you can just speak out. But I was also thinking, my initial question about what's the right tool was also, is there an easy way to search for tools? Is there an inventory?
Yeah, and that's something that people new to genomics really struggle with, because there are kind of, I guess, repositories, and then there's tools like if you have Galaxy or if you use Scienverse, but they tend to be, I guess, software agnostic. They're not saying, if you want to do a SNP call, you only use this tool, but rather they do is they take all the open tools, make them available, and then say to you, you get to pick which one you want to use. That's great for people who know what they're doing and know what they want to use, but it's much less helpful for people who don't know where to start. And common questions I get with people who are just starting is, what even are my options? How do I find out what's available? Right. And that's not an easy question to answer.
Yeah. I can see that it very quickly becomes very confusing for people new at this.
I think that one of the number one, if you ask people why they're selecting the software that they are, you'd like to think that there was some high-minded critical thinking about that, but really what it is this is the workflow that I could get to work and I could understand, and that's it, right? And that's the problem.
Yeah. So, and there's a comment here from Maria. I don't think there's a question in there, but she appreciates the the workflows and the presentation. So, any other questions for Fiona or for Daniel? I know.
I see there's a question here about the bioinformatics workshop materials.
Yes. Okay.
So that's already been held, but if you're interested in contributing to those discussion questions, that link is there, that poll is open. Just so you know, Mashoud, that we're trying to get to the point of, like I say, synthesizing all those many opinions and and the information we get, and building that into a coherent report that we will make public as soon as we have it. I don't know if that answers your question exactly.
Yeah, I think that will be very helpful to get that. Then there's a question from Nathani. I think it's a good idea to encourage the community to write reviews about methods and how they compare. Yeah. So.
Yeah, I agree. You know, we don't do that very often, right? And, you know, I think those make great student projects as well. There's a lot of learning involved in those.
Yeah, yeah. I mean, that part of the problem is that it's such a fast-moving field that, you know, by the time you get that review published, it's out of date, right?
Oh, yeah.
Yeah. Okay. Now I know Daniel has been very busy in answering questions in the chat, so thanks for that, Daniel. And I think you probably have captured most of them, right?
Still working on a couple, but
Okay. Any that you want to address here? We have a few minutes yet. Otherwise, it's fine to just put them in the chat, and then it's going to be saved, and then you can reach out to the individuals. But
sure.
Any last questions, either for Daniel or for Fiona?
There's a question here, and Anna, I think you make a good point. You know, talking about, you know, when you compare two workflows and get different results, then how do you look at accuracy? And, you know, there's a lot of work done to, you know, to do these precision and accuracy and recall type things. But in a lot of these comparisons of these workflows, we simply don't know what the right answers are. And so it's hard to even get the metrics to sort of figure out how to talk about these. Yeah, I agree. So is there an established validation method? I don't think there's going to be a validation method because we don't have a truth set, but at least what we can do is start with a set that works and build from that and say, this is what you at least know what you get out of this set, and you know that this set works.
Yeah, yeah.
It's only a partial answer, Anna. So.
Yeah. Okay. I think we'll close it off here. So, Nicole, can you put up the last slide? So I want to thank Daniel and Fiona for excellent presentations, very informative, and for sharing your results. Also need to thank USDA NIFA for the funding of all of this, and sure many of you will be aware that so NIFA took over the funding of this program last year, and they have the new RFA is also out for this year, and the deadline is May 30 of this year. So have a look at that on the USDA NIFA website. All the information is there, and you know, contact the program leaders if you have questions. For this AG2PI program that we ran here at Iowa State with others, we still have a couple of things that are finishing up. We have another field day on April 24, so another two sets of coconut grant awardees are going to present. And then there will be a workshop, which actually I think ties well to what Fiona was talking about. Workshop on scientific computing and data analytics, a comprehensive toolkit for research, which is also going to be presented by graduate students and undergraduates, actually from the University of Arizona, most of them involved with Doug Pauly's lab. And that also will include some real-world applications related to plant phenotyping. So actually connects to both of today's talks. So if you're interested in that, there's more details on the ag2pi.org website, and you can register for that and register early because they typically fill up fairly quickly. So with that, I want to thank Nicole and Eddie for the logistics and the technical support as usual, and then everybody for attending. And this is being recorded, so if you think this might be interesting to others, the recording should be available in the next couple of days on the AG2PI website, and hope to see you again next month. So thank you all.